Wow. All right, good morning, everyone. In the interest of time, I'm just going to go ahead and get started this morning. For those of you who do need to record attendance, you can text 6F56D to 336-793-9317. I'll give you guys a couple seconds to do that. All right. And I have the pleasure and the honor this morning of introducing Dr. Stephen Krzyzewski. He did his undergrad at the University of Chicago. He got his master's and PhD from UNC from their Department of Epidemiology. He's a professor here in internal medicine and translational research, where he is the co-director of the Stick Center for Healthy Aging, as well as the Associate Dean for Research Development. I could go on and on about his titles, his publications, but we would be here all day and he would not have the opportunity to speak. So I'm going to bring him up and allow him to come forth and talk about, can targeting human biology improve human health?
Thank you very much. Very happy to be here and invited and share some of the things that are going on in the Aging Center with our own. I've been here coming on 17 years, and it's been wonderful. It almost seems like I just got here in some ways, and some ways it feels like I've never been anywhere else, but all good, all in a good way, just so you know that. So there's been a lot going on in aging research, especially in the last 10 years, I'd say. I started doing aging research in about 1995, and we got to start a big cohort study called the Health ABC study. And I remember asking myself at the time, do you think we'll ever have a pill that can do anything about aging? And I said to myself, no, probably not in my career. And I don't think that anymore. And that's what I want to talk to you about, a little bit about where we are on a journey that might lead to improving the aging of older people or the health of even younger people by targeting the biology of aging directly. So here are my disclosures, especially the best slides here Jamie helped put together. She's, in addition to a great scientist, she's also a very talented graphic artist, so I just want to give her credit. And I'm going to cover 4 things today. The first is a little bit about what we hope to achieve in our aging research. It's not that everyone lives to 200. It's that people have the opportunity to have the best lives that they can for their entire lifespan. We think about that as aging well. We have a rapidly aging society, and so the medical care system is not adding capacity at a rate that will match the demands it will be on it based on how our demographics are changing, and talk a little bit about strategies to address that issue. The name for targeting the biology of aging to help health, the people in that field have decided to call it Geroscience, so that's what I'm going to call it too. So if you haven't heard that word, you're going to hear it more and more, this idea of Geroscience. And then thinking about the future, where we go from where we are now. So how does one age well? Assuming that is, and I will put it, that's really the goal of everything, is to have a fulfilled life at every stage of life. And I break it down into 4 domains. Disease avoidance is good. I think everyone would agree. And that is really the primary focus of the medical care system. In geriatrics, we really focus on functional independence, because as much as people want to remain disease-free, they really want to be able to be independent in their lives and not depend on other people, whether it be for cognitive failure or physical failure. In areas that the medical system isn't quite as comfortable in addressing, social engagement, the ability to fulfill in a meaningful way those roles that you have with others and your ability to engage with others. And finally, meaning and purpose in life. So I would say these are not traditionally the domain of the medical care system, but as we think about helping aging populations, we might need to consider how to incorporate some of these social ideas about aging well into our medical care models in some fashion. So here's the first, so this is Grand Rounds, you're getting CME credits for this, so here's your first test. Do you suppose this man will age well? Okay, no one's answering, you don't get any credit for this. No, I'm seeing no. I kind of agree with you. So what do you see here that makes you think he's not going to age well? Yeah. In this one slide, basically summarizes the past 20 years of Sick Center research, all the things that this guy is doing, we try to get people not to do.
Or put it otherwise, what he's doing is preventing old age.
Yeah, in a way, right. In a way, he's preventing old age, right. So I was at a meeting, and we were talking about this, and someone said, are we ever going to have a pill to stop the aging process? And I said, yes, cyanide. So anyway, so yes, so this person is clearly sedentary. He's indoors, he's obese, he's eating chips over here. He's not interacting with anybody, he's completely alone. He's in the dark, he's not getting sunlight, and there are probably several other things that one could identify about this gentleman that would suggest he's not going to age in the best possible way. So out of all the research we've done in the Stick Center, at least from the standpoint of independence and the standpoint of disease avoidance, we can say if you maintain caloric balance, that's good. Dietary patterns may be important. Exercise and moving around, I'll explain the difference in a moment. Managing cardiometabolic disease and staying engaged and involved. And I'll show you one data slide for the first four of these, give you an idea what I'm talking about. So this is data from the Women's Health Initiative Outcome Study, which had 36,000 women in it, and they took everyone when they came into the study and saw what their body mass index is, which is a measure of obesity, and we say that 30 is the cut point. Anyone higher than 30 is considered obese, and 25 to 30 in some documents is referred to as overweight. The desirable weight is 18.5 to 25, and less than 18.5 people are considered underweight and likely possibly malnourished. So everyone coming in, what were they like when they were at 85? Well, a lot of women did not live to 85. And you can see both the underweight and the highly obese women were much more likely to die and never make it to 85. Incident disease, which women develop new chronic diseases over the follow-up period, and this is not counting the women who came into the study already having established disease. And you can see at every level of obesity, there were increased odds of acquiring a new chronic disease. So I'm very interested in mobility disability, and this is very striking. Any level of overweight, especially obesity type 1 and type 2, highly associated with mobility disability. So we study a lot about obesity in our center because of its incredible impact on the mobility function of older people, which is something that we're interested in maintaining so older people can maintain independence. So all the information about dietary pattern and health and function is all observational. It's all from epidemiologic studies. And these are several on the references on the bottom here. And I had to prepare a handout with references so it's available somehow to you of several studies looking at various different patterns than just going to McDonald's and eating fast food all the time. And I just bring this up just to say there's some common patterns. A lot of fruits and vegetables, whole grains, de-emphasizing of red meat and processed meats, in moderate alcohol consumption, actually, in a couple of them, low sodium, low sweetened beverages, So this is an index developed by the people who do nutrition research at Harvard. Mediterranean diet is based on the pattern of diets of Mediterranean peoples of very, very low rates of certainly metabolic disease and good function late in life. And the DASH diet is a diet designed to prevent hypertension. So basically the story is, don't eat like advertisers want you to eat. Eat like this, any of these, and your odds of maintaining function are enhanced. So we are the #1 recommendation for every person in aging about how to maintain your function is to exercise. And there are hundreds of studies showing a number of benefits of exercise. We're learning more recently there's not only exercise, but it's the pattern of movement through the day that might be interesting. So this is a summary of 1 million people and their experience. And these 4 groups on the bottom, you see the Met hours per week, are quartiles of moderate to vigorous physical activity in a week. So the people on the extreme right are very not vigorous exercising. The people on the left are exercising quite a bit. And then the little bars, the little dots are for sitting time during the day, and it goes from less than 4 hours to up to greater than 8 hours. And what you can see is that no matter what category of maximum physical activity or moderate to vigorous, the more you sit, the worse off you are. And the being very vigorous in exercise blunts that effect, but doesn't ever go away no matter how much that you're doing. We just had at the Stick Center, Jason Fanning, who's an expert in this area, present, and I was distressed to learn that standing desks don't fix this. So unless you have the desk going up and down all the time, that could help you. So in terms of managing metabolic risk factors, I think we should applaud. I see Dr. Williamson here in the Sprint Mind study, which was a study of typical blood pressure control and intensive targets. And if you're here at Wake Forest, I can't believe you wouldn't have heard about this study already, because this study was noted by JAMA as one of the 10 most important articles they did in the last decade. So, and Dr. Williamson led that. And it's really the first study that shows that if you intensely control blood pressure with lower targets compared to regular control, you actually reduce the rates of mild cognitive impairment, which is a pre-dementia state, or the combination, a composite of mild cognitive impairment and probable dementia. And the reason this is important is we've had lots of data showing that metabolic problems predict Alzheimer's. This is the first trial that shows that actually intervening on the intermediary can actually prevent the outcome. So it's incredibly important. So what are our strategies for an aging society? And sort of all of pharma, mostly, pretty much, and a lot of medicine is arranged around this idea, is that we target one mechanism for one disease. So cholesterol and cancer, we target cholesterol. Blood pressure and stroke, we target blood pressure. There are a number of examples of this idea, and FDA is actually set up that way. It's the easiest drug approval is a specific mechanism for a specific disease. You drug the mechanism, you change the outcome. FDA just is fine with that. Their approval system is very much tuned for that approach. So these are the risk factors for heart disease, hypertension, smoking, diabetes, cholesterol. And this represents, so this is a slide lent to me by Steve Ostad, who's a leading gerontologist who's now at UAB. And so this is a % increase in risk of heart disease for people who are in their 40s compared to people who are over 60. And so this is the accumulated excess risk for people with these risk factors over this, between those two groups. So we'd all agree that, well, we should stop smoking and we should treat hypertension, and all those things are true, but it neglects the most important risk factor, which is age. Because and so if you plot age on the same axis, there's a 66-fold increase in heart disease just due to age that has nothing to do with the risk factors. So, and in fact, if you look at death rates for heart disease, stroke, cancer, pneumonia, a number of conditions, they all rise exponentially with age. And it's not linear. And this is a problem because if we prevent cancer, people are still exposed to exponentially increasing risk of everything that's not cancer. And if we can prevent diabetes, people are still exposed to exponential increasing risk of everything else, and that's because they're aging, not because of the common risk factors. So that would play a role. I'm not going to diminish that. So wouldn't it be great if we could do something about aging per se? In addition to diseases, and this is where function comes, the aging well is disease avoidance, but it's also functional maintenance. So as we age, and this has nothing to do with a specific disease process, because these are physiologic changes that are observed in people longitudinally regardless of the underlying health condition. Their metabolic rate goes down, their heart rate goes down, muscle strength goes down, bone mineral mass, vital capacity, VO2 max. Also, and these are longitudinal data, mostly from men from the Baltimore longitudinal study, which was started in the 40s and has followed people from their 20s to their 60s by now, and people in their 60s till death by now. And this represents sort of the plotting of all the physiologic changes from youth to old age. And function, our independence, our environment depends and assumes a certain level of VO2 max. And if we can't generate that VO2 max, we can't live in this environment. So a lot of what we define in the gerontologists call disability is when your physiologic capacity does not match the environment that you're in. And so imagine yourself as a hunter-gatherer having to go pelt down mastodons in order to live. I would bet almost everyone in this room would count as disabled because I'm pretty sure most of our capacities don't match that environment very well. So we're privileged to live in a time where we can be productive and contributing as many of our physiologic capacity, much of it declines. So here's the next CME part of the talk. What do these organisms have in common? Yeast, nematode, fruit fly, and mouse. And because we have to finish by 9, I will tell you that these are each model organisms that have been used to understand the biology of aging in different ways. And one of the important and impressive findings that there are mechanisms that are related to the aging of all of these organisms that seem conserved across these incredibly diverse phyla. So there are there's a common biology of aging, so we can actually learn stuff from yeasts and worms that may have some implications for human health. So, and these models have shown us not only that there's a common biology, but that biology can be targeted. And there are many ways. So most of the worm and yeast work generated just lots of mutants and looked at long-lived mutants and saw what areas were mutated. The mouse and fruit fly, also a little bit in the mouse and fruit fly, but in mouse it's sort of what kinds of interventions can we do to extend the life of the mouse. One of the things that works in all these is caloric restriction. If you don't give these animals as much nutrients as they would like, they live longer, and their health span is increased. So lifespan is how long the animals live. Health span is how much of that life is disease-free. And I don't really know how you find health span in a nematode, quite honestly. because it's really hard to take their temperature, and the stethoscope doesn't work. But the mouse, they do a pretty good job and can document various things, and both lifespan and health span is increased. And what's come out of that work is a recognition of what some people call the hallmarks of aging or the pillars of aging. And there are a couple of very good reviews. This one gets cited a lot because they have a really pretty picture in their paper that everyone likes to show as a slide, so I am doing that. And these are the areas that are most affected or routinely affected and conserved pathways that are involved in the aging process. And I'll read them quickly because it's a little probably hard for the people in the back to see this unless you turn around and look at the back screen, and don't do that. I'd feel like you're telling me something. Genomic instability, telomere attrition, epigenetic alterations, loss of proteostasis, deregulated nutrient sensing, mitochondrial dysfunction, cellular senescence, stem cell exhaustion, and altered intercellular communication. So these are revealed both by mutations and targeting and siRNA. All the tools of molecular biology and manipulation have been used to identify the roles of these various pathways. They're presented as pillars, but none of them are really independent of each other, and they all interact. And if you push down on one, it will affect several of the others. A lot of the early work has pointed to insulin signaling, IGF-1 signaling as a key pathway in all of this. So that the fact there is a common biology that might be targetable provides an alternative strategy of what we might do in aging. So the current, I call it the FDA way of thinking, is to delay specific disease through controlling specific risk factors, and that really works. Actually, I mean, statins are great at preventing heart disease. But if you look at the data from statin trials, that is exactly what they do. Their all-cause mortality from everything that's not atherosclerotic really isn't affected very much by statin use. So the new Geroscience paradigm is that we can delay aging by targeting basic molecular processes of aging and perhaps delay multiple diseases all at once. So that seems like a good thing to do. And, you know, as usual, the world out there is a little ahead of academia in some ways. There was a couple of years back a documentary series sponsored by National Geographic, breakthroughs, things that were going to change the next decade, and there was one on water and one on fusion. There was also one on this idea of geroscience, the age of aging, and it was, this particular documentary was produced by Ron Howard and his production company, and it tells this early part of the story of people, really a project I'm going to tell you about in a few minutes, called TAME, about this idea, can we address aging per se as a way of preventing multiple diseases? So this is not crazy. This is where another role of the mice is they provide a convenient and relatively inexpensive way of testing different compounds to see if they actually affect the biology of aging. And there is a very well organized enterprise that the National Institute of Aging funds called the Interventions Testing Program, where they have 3 sites. People out here, you guys, can nominate compounds. They have an evaluation process, and if they like your compound, they will give it to hundreds and hundreds of mice, both males and females, at 3 different locations, usually starting at about 4 months of age, sometimes starting later, and they see how long it takes for the mice to die, and basically see where there's evidence in a mammalian model that you can actually delay aging. So they've actually, I have 7 out of 16, I think it's really 7 out of 19 or 20 compounds they've tried so far, but they've had 7 that have shown some benefit in either male or female, or in at least one sex of mouse, okay? Mostly it's the males who are helped, because I hate to break it to some of the people here, but men don't live as long as women, so maybe there's opportunities for interventions in males that maybe women don't need, perhaps, I don't know. But these are some of the things that have been tested. The current sort of champion, I think, in the minds of the aging world is rapamycin, which is an immune suppressant drug. And it was the drug that had the best effect in both males and female mice. So most of the findings have been mostly in the male mice, but rapamycin was in both. And I'll tell you the importance or the role that rapamycin might play, because the rapamycin and its targets have been a major story in the biology of aging. So now I'm going to tell you about 3 Geroscience vignettes, 3 attempts or versions of applications of these ideas of the biology of aging to human health. And I'll talk first about senescent cells, then telomere shortening and telomerase, and then the prospect of targeting mitochondria as a target for enhancing function-enhancing therapy. So I'm not surprised, but a lot of people really haven't heard much about senescent cells. It's a cell fate that doesn't get discussed that much. So you can have a cell that's in your body just doing what it's supposed to be doing and replicating happily. If that cell gets damaged, it might die. It might go through a programmed cell death program called apoptosis. But there's yet another fate. It could become senescent. And many of the treatments that are used, especially in oncology, actually are used in model systems to induce senescence. So both radiation and DNA intercalating agents are used by cell researchers to make cells senescent through damage-related mechanisms. So what happens? So there's some kind of DNA damage. These are 2 key pathways in maintaining and inducing senescence. There's a growth arrest. Gene expression changes. Those cells become resistant to apoptosis. And depending on how they're damaged and how they become senescent, they can also start secreting a fairly toxic stew of cytokines, chemokines, and growth factors that can have paracrine effects on the surrounding tissues to really screw things up. You can genetically modulate mice to have a big senescent burden. And if you could turn that mouse into a person, you say that person is frail. The burden of senescent disease leads to a series of behaviors in mice that look very much like human frailty. They accumulate in various tissues, and and we're hoping that if we can clear those, we can mitigate the effects of the volume of these cells and the things that they're secreting in the body. So I'm going to tell you a story about a project that we did here that Jamie Justice did most of the work on, and Rudy Pasquale also was heavily involved in. And it's a partnership with the Mayo Clinic and Jim Kirkland's group and the UT San Antonio and Nick Musey's group. And they the basic biology of senescence, one of the epicenters of that is the Mayo Clinic, and there are some competing groups up there, but Jim Kirkland was looking at what is it about senescent cells that keeps them from dying, and they did comparative genomic scans to differentiate the key expression networks that seem to be preventing senescent cells from being knocked off in the body. And I'm not going to explain that network because I couldn't possibly, only to say that they were able to come up with networks and then use another database of drugs that have already been approved for human use that might attack or target some of the nodes in this network, and use that as the starting point for drugs that might affect the burden of senescent cells. And they found 2. And there are several, but 2 were attractive. One is dasatinib, which is a drug that's used in treatment of certain leukemias. And you can read, it's a tyrosine kinase, and you can read a little bit of the attributes there. The other is a polyphenol that's found in onions and apple skins and a number of places called quercetin. And in model systems, in cell systems, these will induce apoptosis in senescent cells. The dasatinib in preadipocytes and quercetin in senescent human endothelial cell progenitors. And so you take that to the mouse, and you can engineer mice to have high senescent burden, and this combination does knock down the percentage of cells in those mice that are senescent. And the thing that you need to realize is this is just one dose. It's sort of like if you're trying to eradicate cancer and you have a pill that knocks all the cancer out, you don't keep giving them those pills. You're done. And so these targeting strategies only have to be done once or twice to knock down the level of the cells, and you should see the benefits. And that's what you see in the mice anyway, where you see improved treadmill endurance, symptoms of frailty, better cardiac and metabolic function when you knock down the burden of these cells. So who would you try this in a human study? And through some thinking of the group, and the thought was, we'll start with idiopathic pulmonary fibrosis, which is a terrible disease. Someone is probably leaning against the dimmer switch, so that's good. Okay, so it's a terrible disease. Since we don't know that, I mean, senescence is involved in some normal processes like wound healing, and might be a way to keep people from getting cancer. Maybe that's, and people do believe that one of the reasons we even have senescence is a way to keep us all from dying of cancer at 30. So I don't know if that's true, but those are what some of the thinkers in the aging world think. So you wouldn't want to give this combination to just anybody. The cost-benefit analysis, so you should really give it to folks who are any benefit would really be appreciated, and they're willing to withstand a little bit of risk tolerance because of the kinds of patients they are. So the decision was to go with people with idiopathic pulmonary fibrosis, and their lung tissue looks like a place where there'd be a lot of cell senescence, and in fact, if you stain lung tissue for senescent markers, are very common in the lung tissue of people with IPF. And so there's a strong sort of a phenotypic phenotypic correspondence. So there is a model of this where a bleomycin-induced IPF model, and this is the experiment where they gave these mice, DQ is the combination vehicle as the control group, just on 3 occasions over a couple-week period. And so the color says the IPF plus DQ is in orange, but in both figures it's actually in yellow. So it's a little confusing, but what it shows is that the pulmonary function measure in mice is rescued in the IPF-induced model when you also give them DQ, and their running distance is recovered when they have the IPF model and D plus Q. So we were proud to be able to publish the first human study, and this is just an open-labeled safety-based intolerance study. Here, the people in the group are all represented on this slide. And what we did was a proof of concept, recruited folks both here and in San Antonio with IPF, and basically gave them 9 doses of this combination, 3 Monday, Tuesday, and Wednesday on 3 consecutive weeks, and it was a month-long study just to see if this is tolerable. The folks at San Antonio are going on to do a little randomized version of this just to see if some of the functional improvements that we saw might be because of failure, because of lack of placebo control. So the very early days, like I said, this is the first human publication, and I don't think there have been any others since in this, but this is what we saw. We saw improvements in a 6-minute walk, SBB score, time chair rise. And again, this is open label, no control group, so this could be a training effect easily. I would observe, though, that the standard of care for IPF patients is a 6-minute walk. So our study wasn't the first time they ever had a 6-minute walk. So we're wondering, but it's enough for us to go forward with. And we really didn't see any differences in this very small sample over this very short time in a number of other kinds of things. But it seemed completely safe. All the side effects were exactly as we expected. Nothing lurking there that popped out in that small number of folks. Okay, so my mother was a big fan of this area, not in a good way. And so she passed about a year ago. I'm getting all her forwarded mail, and you can't believe the kinds of stuff people are trying to sell older people who want to live longer or look better. And so they take little bits of the popular press to legitimize themselves, and this is from one of the mailings. One capsule can diffuse the telomere ticking time bomb. And so there is a woman who is presumably the and here's the end test. And the fact there are different women should not get in your way of appreciating this, okay? So telomeres are on the caps of all the chromosomes. And every time you reproduce a cell, those get shorter. And if telomerase isn't making them longer again, if they get too short, then the cells can't divide anymore. In fact, this is really probably the very earliest observation in the biology of aging was in the early 60s, and there's a famous gerontologist, scientist, Leonard Hayflick, and he was trying to culture cells, and he could never get them to replicate more than about 30 times. And that was called replicative senescence, which is different than damaged senescence. They're interrelated, and it became known that the reason there was replicative senescence was because of this telomere shortening. So of course it makes perfect sense that we should do everything we should possibly do to have the longest telomeres. I will tell you in this part, when we look in epidemiologic studies of older people and try to either characterize their telomerase activity or their telomere length, we don't see very much. So the people within the field of aging research have sort of said, well, this is interesting, a clearly important biologic mechanism, probably more important in cancer, because the screw up of this system is what helps cancer cells be and remain immortal. But in normal aging, probably not a great target. And I want to show you this one piece of data because it's really a cool study and is sort of interesting in a couple of ways. So this is a great study called the UK Biobank. So the folks in the UK have gotten organized to enroll 500,000 people and bank all their samples and follow their health histories through the National Health Service and collect other kinds of information on these folks. And one of the things they did was characterize their genome for all the mutations that are known to either lead to longer or shorter telomeres. And they add them all up, and they use 13 SNPs associated with telomere length. So these are people who are born and live their whole life with either relatively longer or relatively shorter telomeres. So you'd guess if telomeres were the answer to everything, the people born with longer telomeres should live longer. When you look at all-cause mortality, there's no difference. There's no difference by this. So they were able to estimate estimates for development of diseases or conditions with a predicted 250 base pair difference using this genomic, functional genomic approach. And so I'm not going to go through all the little dots over there. Those are the forest plots for all the things I looked at. But distressingly, the people with longer telomeres had a higher risk of cancer, a lower risk of heart disease. So that's one of the reasons you don't see all-cause mortality differences. They kind of net out. And interesting also, the people with longer telomeres had higher systolic blood pressures and better pulmonary function. So FEV and VEC is different. So I want to emphasize that the aging of biology is normal biology, and it's going to be a little bit hydraulic because we're going to distress one thing, there's going to be compensation. And this might be an example of what's good for one class of disease might be bad for another class of disease. So that's a very important sort of concept out of all this, is keeping our eyes open for that possibility. So the next piece of this is about mitochondria, and this is, Wikipedia is wonderful, and there's a lovely picture, and mitochondria, we study a lot about mobility and muscle function, so we're really interested in the mitochondria in and around the muscle as a supply of energy. But it probably affects a lot of clinical populations in ways we don't quite understand. This is an example of that. So your mitochondria has its own DNA. It does not have a DNA repair mechanism. So there's no way for mutated mitochondrial DNA to get fixed. And there are inherited mitochondrial diseases, and one of them is a set of diseases is based on the inheritance of this one particular mutation, A to G mutation, on one thing. And it expresses very differently in the people who inherit it, and it can lead to something called knee loss or deafness or myopathy, cardiomyopathy, cognitive impairment. And it's not the same in every person, and so it's quite an interesting kind of thing. As we age, our mitochondria accumulate mutation, and the extent of that accumulation is referred to as heteroplasmy. And so in a study that I worked with, a colleague of mine, Greg Trenah, took the white cells from about 1,000 people in this cohort study called the Health ABC study and characterized their mitochondria for heteroplasmy at this location that's associated with inherited mitochondrial disease. None of these people had inherited mitochondrial disease, right? They only developed mutations at the same site that those people had. And what you saw was a pleiotropic set of deficits which mirrored the kinds of things you saw in the people with inherited defect. So, for example, fasting insulin was much higher in the people who had a lot of, so these are tertiles of heteroplasmy. So almost none, 4% or less, 4 to 6% or 6 to 19%. They were also weaker. They also had poorer scores on a digit symbol substitution, a cognitive exam. They also had much higher stroke mortality and much higher and somewhat higher all-cause mortality. So the same kinds of problems you saw in this inherited disease, people accumulated similar flavor of damage as they age and led to the same constellation of deficits. Now, this is a challenge to medicine because this does not fall neatly into any anatomic system, which is the way medical schools are organized and think. So this is definitely cross-cutting, and one of the opportunities of a geroscientific view of health that might be a little bit complementary to the traditional anatomic view of health. So this plays out in another way. So this is Delane Kitzman published a study of weight loss, which is the D, the diet, exercise, aerobic exercise, in people with heart failure with preserved ejection fraction, and many of these people are overweight and obese. And if you look at their change in peak VO2, which is our most sensitive measure of aerobic function, exercise, they can train up a little bit. Weight loss actually improved aerobic function, and the combination of the two added, okay? So this group had, the D&E group had the most improvements in quality of life, more than the exercise group alone. Working with Anthony Molina, when he, who recently left to lead a program in San Diego, they looked at muscle biopsies in these groups and looked at porin intensity, and so porin is a measure of mitochondrial mass. And the D&E group, the weight loss actually led to an increase in mitochondrial mass in the individual muscle cells in these folks. And if you plotted the VO2 max delta against the poor and delta, they were also correlated. So mitochondria is a target to improve function in older adults. So what about the future of this? So we're in kind of this is what we hope. So these are actual accumulations of comorbidity with age. These are from a study in Mayo Clinic in Rochester, Minnesota. So we can target aging. We're hoping we can deflect that line. How would you do that? So the model at the interventions testing program starts with a 4-month-old mouse. Okay, a 4-month-old mouse is equivalent-ish to about a 40-year-old person, and then they follow them all till they die. We're never going to do a 50-year study of a preventive strategy in humans. So there's this whole continuum of things that we can look at, changes in all the way from gene expression and cellular physiology that we can do easily to lower mortality rate, which in some way is the proof of the pudding to some extent. And interestingly, in this arena, FDA is only interested in diseases and death. We had a delegation that went to interview with FDA, and one of the FDA people in the room said, well, aging is a philosophical concept. It's not something we're authorized to approve drugs to treat. It's not a treatable, it's not a condition, and it's not a disease. And so we're kind of at the standoff of people trying to target aging pathways, but there's no FDA pathway to get any of those approved, so, or relatively few of those. So we are hoping, and I see Mark Esplin, who's been a major contributor to this effort, and Nir Barzilai and several others are working to, we've designed and have promise of funding for a randomized controlled trial called Targeting Aging with Metformin. And you can see the study criteria here, and we're trying to raise private money, and we have pledges, but no money yet, to actually field this study to show that metformin, whether or not it can or cannot prevent the emergence of additional comorbidities as people age. And if you take these hallmarks and look at the cell science around metformin, you can see pleiotropic effects on many of the hallmarks that are attributable to metformin. And importantly, metformin has very important effects on insulin signaling and IGF-1. If you're interested in aging, the SIRT system and mTOR are two very important pathways. mTOR is molecular target of rapamycin. So we talked about rapamycin early. That is a way to manipulate that, and the downregulation of that prevents growth, but it also seems to extend lifespan and health span. So these are some one mouse experiment where they started metformin already in the old age of those mice, already over 40 weeks, and saw a 6% expansion of lifespan, but a number of health span benefits in those mice. And much of the evidence in humans is epidemiologic. It's comparing people with type 2 diabetes who take metformin to people with type 2 diabetes who take other things. So it's not super great evidence, but it is pretty consistently beneficial. There is randomized clinical trial evidence that shows that metformin in people with very early diabetes does prevent CVD and all-cause mortality, and it certainly has been shown to prevent diabetes. And these are sort of representative medians of the sample size. But we admit there are negative studies, and sort of at a state of equipoise, but it's very safe and very inexpensive. And if you're ever going to try a prevention study, it's got to be safe and it's got to be cheap, because we don't want to be putting people at risk of anything on a flyer, right? We want to have something we know is likely to be very safe for them. So I don't know if you guys have, in your first year, had probably seen this yourself in a microscope. So this is Beverly Hill Lab. This is telolift. This is what an old cell looks like. Did you know that? You know, your pathology books are all wrong, and this is what a young cell looks like. So you want to know how much a 1-month supply of telomere lift would run you? $90. That's exactly right. But if you get if you got a 3-month supply, it's only $163 and $7.95 for shipping and handling for Tela lift for a 3-month supply. So just so you go, and hey, Nobel Prize, can't be bad. Nature magazine. So there you go. So what I'd like you to take away from this is that medicine can contribute to aging well, but many aspects are out of the traditional scope of healthcare. We should just be humble about that, acknowledge that people have values and things they want to do that treating a disease is not necessarily going to address. I think aging biology provides interesting and novel insights in the problems of older patients that may not be apparent when studying disease, focusing on disease-specific mechanisms. And the Geroscience hypothesis, if it's proved correct, and metformin may not be the thing, it might be something else, it will transform population health management and the care of older folks. And we're pulling together a affinity group around Geroscience, and we're going to meet for the first time on the 27th, Monday at 3. And if you're interested in being on that mailing list, let Jamie Justice know, and she's sort of managing that list right now. So, and that's the stuff that I was told to add. So thank you very much.
Does anyone have any questions?
One of the attributes of a good death is that it happens quickly. So there is a philosophical issue of you want to live well forever and then die quickly.
Right.
Would this tie in with that, the second?
I think so. I think there's a famous paper from the 80s, I think, called The Compression of Morbidity. And the idea, even the stuff we're doing now, has led to the pushing of morbidities closer to the end of life, and so you've got this curve where you're living and you die, and it goes like this. For much of human history, it was sort of a sloping downward, very high early mortality, and then constant erosion of that, and almost sort of a hyperbolic loss. And as we've gotten better at keeping moms and babies alive, we're keeping many, many more people alive till their 60s, and that's when all these age-related diseases start occurring. We're hoping, the idea here is not to get people to live to 120. It really is to extend the health span and compress the diseases that help make life miserable, and compress those as far to the left of that curve as possible. So we don't know if that would work. That's true in animals, and it's also true in the offspring of centenarians. So a number of centenarian studies have looked at their offspring, and their offspring develop chronic diseases later in their chronologic age than their spouses do. So there is some evidence that, these have to be related. When you die, so here's a question for everyone here. Why do you die on a given day? Right? There's no really good explanation. Why it's Tuesday and not Friday? Or why was it this year and not? You have all the same chronic disease, all the same thing, and one day you die and one day you don't. And it's a little bit, my theory, or the way I view it, is you've got all these things wrong, and sometimes that constellation lines up, and it just happens that particular configuration of problems just can't support life, and that's why you die on that day. You got unlucky on that day. And so if we stop all those things from happening, we're reducing the chances of death, but we'll also reduce the things that are going wrong up to the time of death, and that will help compress, increase the health span. So that's how I think about it.
Thanks a lot, Steve. I was wondering if you could comment briefly on your interactions with the FDA. The FDA seems to have a not consistent message for how they approve certain drugs for certain indications, and just in the context of TAME and sort of what you're envisioning as the primary outcome of that study, and how the FDA sort of weighed in on this and what they might approve the drug for if X or Y is hit.
Right. No, thanks. I should have mentioned that. So saying what does the FDA think is like saying what does the government think. It's a very big organization, and it's a rabbit warren of various things. There's an endocrine division, a neurology division, a little bit of what they approve and how they think about it. Depends on what division you end up in. So our interaction with TAME involved people from many of the divisions, and including Bob Temple, who was sort of the senior scientist, approval scientist at that. And so we went in there saying, we would like, to prevent aging, and what would that look like? And so the conversation, well, aging isn't a thing. How about if we prevented age-related diseases from occurring? And basically the message was, yes, now you're talking our term. We're very interested in things that can prevent disease. That's something we are authorized to license. But don't include diabetes in your list. And one of the persons said, so I probably should turn this off, but said, well, that's just a biochemical diagnosis. That's not really, So they weren't as impressed by biochemical or physiologic diagnoses as they were by actual clinical events of clear-cut clinical relevance, like cancer, Alzheimer's, heart disease, stroke, heart failure, hospitals. And so they didn't come out and say, yes, they would approve a drug to prevent this constellation, but they said, we can't, that's the only, the implication was they needed hard clinical endpoints early to really even make it mean anything to them. So it didn't, it's like doesn't even register, it's not even wrong unless it's entities that they are authorized to register and license drugs around. That's how I took it away. I know. Mark, is that generally confirmed with your view?
We had some interest in wrinkles.
Oh, they did, yeah. They really pushed us on the wrinkle part. They said, you sure you can't do anything about wrinkles? Yeah, they were really, they said, then you've done something about aging. Wrinkles and gray hair. If we could do something about that, they would have been really interested in that.
All right. So I think for the interest of time, we'll thank you, Dr. Constitutes.
Thank you. And your name was, I missed it. Ryan, I heard that. Thanks for organizing this.